Okay, let's get started. My name is Peter Duncan, and I'm the Director of Product Management here at Codex. And welcome to our webinar, Harnessing the Power of Synthetic Biology to Combat COVID-19. Today, I'm really excited to present three amazing speakers that will all discuss the highly relevant topic of global pandemics, including COVID-19, which we are all currently experiencing together. Dr. Dan Gibson will lead off and discuss how synthetic biology has been used in the past for fighting pandemics and how it can also be utilized today. Next, Katie Lyons will talk about how DNA printing can accelerate vaccine research and development for current and future pandemics. And then finally, Dr. Krishna Kannan will present how we synthesize the world's first SARS-CoV-2 complete full-length genome de novo and discuss real-world applications of this work. At the end of the webinar, we will have a Q&A session where we will do our best to answer what I'm sure will be many questions. It is my pleasure to introduce our Chief Technology Officer, Dr. Dan Gibson. Dan is one of the most influential thought leaders in the field of synthetic biology, and here are a few highlights. Just recently, Dan and his team here at Codex have created the world's first synthetic SARS-CoV-2 full-length genome, which we'll discuss shortly. In 2016, he led a team that developed the first synthesized minimal cell. In 2010, he constructed the first synthetic cell. And in 2009, he published what is now the most widely used method for cloning, the Gibson Assembly method, which has over 4,000 citations and is the foundational technology for enabling the world's only DNA printer. Finally, in 2008, Dan co-led the team that constructed the first synthetic bacterial genome. Without further delay, I am pleased to pass the controls over to Dan.
Well, thank you, Peter. Hello, everyone. Over the past 15 years, my teams have been developing widely applicable methods for whole genome design, synthesis, and assembly. At Codex, we are focused on providing researchers with those tools to rapidly design and synthesize high-quality DNA to accelerate advances across several fields, including personalized medicine, drug discovery, and vaccine development. In 2010, all of the technologies that we've developed over the past 20 years for reading and writing DNA converged when we announced the creation of the first synthetic cell. Ever since the sequencing of the first bacterial genome in 1995, thousands more complete bacterial genomes have been sequenced and stored in computer databases. The creation of the first synthetic cell was a proof of concept that we could reverse this process. Pull a DNA sequence out of the computer and then convert that information into a free-living, self-replicating cell with all of the expected characteristics of the species that we constructed. This was a project that could not be accomplished with traditional cloning methods and really forced the development of new and advanced molecular biology tool sets. It also paved the way for the generation of synthetic bacteria and viruses, including SARS-CoV-2. Enabling a design-build test cycle at the whole genome level. Our massive efforts to create synthetic bacteria have made us world experts at writing DNA. Throughout these projects, we continue to find ways to write DNA faster, more accurately, and more reliably. And because of the robustness of these technologies, we found that we could readily automate the process to move the workflows out of the scientist's hands and onto a machine. In 2013, we built the BioXP, and it has become absolutely essential for writing DNA across a variety of applications my team and researchers around the world are working on. We immediately realized the BioXP's potential to address global pandemics and started working with a vaccine manufacturer and the US government to bring this to fruition. It was shortly after we built the BioXP that we received an email from an international health organization alerting us that 2 men died in China shortly after contracting the H7N9 bird flu. There were fears of a global pandemic as the virus started rapidly moving across China. A team of Chinese scientists isolated the virus, sequenced the DNA, and then posted the DNA sequence on the internet. At the request of the US government, we downloaded the DNA sequence of the virus and synthesized the DNA in less than 12 hours on the BioXP. And with our partner, we quickly started turning that DNA into a flu vaccine. Meanwhile, the CDC, using technology dating back to the 1940s, was still waiting for the virus to be delivered from China so they could begin their chicken egg-based process for making the vaccine. For the first time, we had a flu vaccine produced ahead of time for a new and potentially dangerous strain, and the US government ordered a stockpile. With the COVID-19 outbreak, we now have a similar situation, but at a greater scale. The major difference now is that our team does not have to respond in isolation. Our BioXP instrument and the tools we created are now available to the rest of the world and has enabled a distributed manufacturing response where scientists can now work collaboratively and in parallel as a community to get ahead of the coronavirus outbreak and generate a vaccine against it. With increased international travel and a population expected to reach 10 billion people by the year 2050, we are going to need innovative synthetic biology solutions for vaccines, diagnostics, and treatments for this pandemic and future disease outbreaks. Our automated DNA printing solutions have great potential in rapidly creating vaccines on the front lines and for specifically tailoring a vaccine to match the strain circulating in a particular area. And this approach can be used for all vaccine types, whether it's DNA vaccines, RNA vaccines, viral vector vaccines, protein-based vaccines, or even live attenuated vaccines. Next, you will hear from Katie Lyons, who is a senior scientist at Codex. Katie has been a part of our organization from day one and worked with the engineers to get the biology running on the BioXP instrument. She will discuss the BioXP and its capabilities. Then you will hear from Krishna Kannan, who is a staff scientist at Codex. Krishna has been on my team for several years and has played important roles in several synthetic cell projects. He will discuss how he used the BioXP to rapidly construct the complete SARS-CoV-2 genome.
Thanks a lot, Dan. So my name is Katie Lyons, and I'm a senior scientist at Codex. And I'm really actually very excited to be talking with everyone today. This pandemic has been such a challenge on so many levels, but in the scientific community, it's been really energizing and really exciting to collaborate with so many people and have the chance to share our technology. So Dan gave a really nice overview of the tool that I'm going to be talking today, and I think of it kind of as a leg up that we have in accelerating research, and it's something near and dear to my heart since I've been working on it for a really long time and working with it for a long time. But I think the real key here is the work that Dan talked about and the work that Krishna has done. All of this can be done without the tool, the BioXP, which I'm going to be talking about. But in order, when there's pressure on, for example, like now, when pretty much every day, every second almost matters, so it's important to use every tool that you have at your disposal in order to accelerate your workflow and do everything as quickly and efficiently and as correctly as possible. So sort of the crux of where we use this machine and the reason that it helps us to move so quickly in a time like this is it reduces friction that can happen at so many points along the way when you're building genes or building genomes. The driver in creating this tool was to make it completely hands-free and to allow both seasoned synthetic biologists who do this by hand all the time and also people who don't to have the same exact experience, which is extremely easy, of loading the machine, pressing start, and letting it do its thing, and out when the program completes, you have your built genes. Over time, we've increased the range of the machine. So when this idea was first sent my way, being a person who builds these things normally, you just think it's not possible. Like, how can you have one protocol to build all of these different types of sequences and be able to, without any input, you know, of checking something on a gel or making sure something works, you know, sort of completely blindly synthesize something from start to back. But so when we started off, this machine was, you know, what we could do in those types of parameters. But over time, we've just gotten better and better, and what was initially a narrow range of constructs is now incredibly broad. We have expanded our complexity and our size and our versatility also in ways that we can approach these builds, and I'll tell you more about that. Additionally, everything is controlled. Again, so if you're working with a multi-channel pipetter, you sometimes don't notice that one of the channels didn't aspirate or whatever. In this case, we know if that channel didn't aspirate, and everything is done exactly in a controlled environment in the same way every time. So you can basically trust the results that come off the machine. We've also expanded the versatility, and we had initially, again, offered sort of a very simple, fragment type of build on these machines, but now we've expanded that to include cloning and a second amplification step to allow you to go up to microgram quantities of DNA. I'll tell you all about that in a few slides. And one of the big things also is that this machine is very supported. So you can be involved as much or as little as you like in how much you know about this machine and how much you interface with this machine. If you know a ton about design and you have excellent prediction tools and you're kind of a seasoned person and enjoy doing it like I do, you may want to just pass something like a sequence to our team and a nucleotide sequence, and that's it. It goes into the portal, it gets sent back to you, and you run it on the machine. But if you don't really want to be involved at all, send us. We have an excellent customer service department. Send us your protein sequence. We can help you here design it. We can do codon optimization. We can help you avoid complexity. There's a whole lot of information that is held in the heads of the people that work here. And then I think the last one is a little bit tongue-in-cheek, but I wanted to say this machine has quite a lineage in that it's the product of the work of people like Dan, people who do this type of assembly day in and day out and do the hardest types of assemblies. We have bioinformaticians who have contributed enormously to allowing us to make these sequences synthesizable by this machine or automatable. We have engineers who are top-notch and work with our biologists. If you want a robot to make your DNA, this is the one to get. And I guess actually it's the only one. So anyway, this is what we do. This is response to pandemics is what we're built for. So it's really exciting actually to be able to kind of present this. So I'll just kind of tell you a little bit more about the machine in more detail. So this is what you'll observe outside of the machine, outside of the box. And again, you don't have to go any deeper than this, but this part you have to know. So you order a sequence, and that, like I said, can be protein or nucleotide. We are able to help you with codon optimization. We have a full database, and we can predict whether or not this machine will have issues synthesizing that particular sequence. And not only that, but we can design around it. From there, you get this nice looking kit, which is a schematic, shown on the right. And so you'll receive a kit that arrives with everything you need to make that. And that's shown in the schematics. Then you take those materials, you load them following a map onto the BioXP, and then close the door, hit start. Most of our protocols take somewhere between 12, actually even shorter than that. It can be something like 8 to about 24 hours for the very longest amplification that we have. But generally, this is something like an overnight run. You start it in the afternoon, and then the next morning it's done for you. So next morning, you work with your genes or clones or amplifications, whatever kit you've decided to order, and you're ready to go the next morning. So all said, that's about 4 to 6 days, and the capacity of the machine is 32 genes per run. Okay, so now a little bit more the nuts and bolts of what goes on inside the machine, if you're interested. So basically, we have taken we patented this oligonucleotide assembly method years ago, and it's really the chassis that everything works off of. Like I said, the starting blocks, the building materials are oligonucleotides. These are then assembled into a double-stranded fragment. But because these are, again, made from synthetic oligos, they are error-prone. They're just randomly sprinkled with errors throughout that fragment. So even though it's built beautifully, it's pretty much not usable at this stage. So the next step then is to, on the part of the workflow of the machine, is to start the error correction protocol. And that is based on, again, I was kind of tooting the horn of our error correction enzyme. So it's a mismatch recognition nuclease, and it's scanning across for bubbles in the DNA where there's an imperfect heteroduplex. So that gets cut and repaired, and then there's actually, I didn't show this on the slide, but there's a second build back step. You end up after the error correction, you end up basically with a big shear profile of DNA, where all of these heteroduplexes have been cut at various spots. So there's a build back step. And then after that, you do have nice material that is error-free and nicely built back in double-stranded format. So the next step, and this is just perfectly amenable for automation, is bead-based cleanup. So we have a magnet on the deck and perfect robot liquid handling. So when we were developing this machine, this was in the early days, a human was pretty much better at every single part of this except the purification, which you just can't beat a robot for doing that. And then, so the last panel there is an example of a gel of, this is 32 genes. They're very similar in their sequence, and you just get this super nice gel, and you have 32 genes ready to go. I will say that this is not always how perfect they look, but in general, we have very robust build back, and this is not unusual to see such a pretty gel like this. So now I'm going to shift gears a little bit and talk about more of the applications of the BioXP. We focused on the last slide about the kind of the foundation of the workflow of the BioXP, which is building fragments. And I just added a little bit of information in terms of how big these fragments can be. I talked earlier about the complexity, so there's a certain threshold and certain parameters that we look for there. But the other one, which is very simple, is just we prefer that the fragments are between 300 base pairs and 1.8 kb. That's the sweet spot on this machine running a fragment style of program. So you can imagine in the SARS-CoV-2 landscape, someone like Krishna, and he'll tell us about this later, putting together all of these fragments to build a genome. So they can be component fragments or they can be standalone, but this is the very foundation of the way this machine runs. But there's more, which is sort of a value added. And another way to look at this is, perhaps most importantly, another sort of bell and whistle attached to this is to increase the hands-free time from gene building and stopping there where the human gets involved to work on downstream processing. But you can extend that one more step by having the machine clone the fragment directly into your vector of choice and end up with a circular circular plasmid that's gone through Gibson Assembly and contains the synthetic DNA that you just built. Additionally, this can be multiplexed so that the synthetic portion can be increased from 1 fragment to 4 fragments and effectively increase that synthetic build size from 1.8 kb maximum to something more like 7 kb. Additionally, conserved sequences can be added between. That would be added by the user along with the vector, and it just increases the versatility of the way that you can use this and maybe more creatively construct what you need to make. And then I also wanted to touch on another way that you can build DNA fragments. They don't have to be one specific sequence. They can be libraries. In this depiction, we're showing a scanning NNN library, site saturation of a specific area of a gene. You can imagine interrogating something like an active site, and the way that the BioXP builds these, we're able to use the repeated sequences in all of the libraries. So what this does is very effectively cuts down on the time it takes to make these libraries. This is the product that runs with the shortest amount of time, roughly 8 hours to complete one of these runs. And additionally, because we're able to reuse so many of those components since they're repeated in all of those sequences, it really effectively cuts the cost as well. And then finally, back to the circular style of building, we have a method called RapidAmp. It's a standalone reagent that's available, but also can be added to the BioXP workflow. This capitalizes on a circular template. So again, this would be something like a clone that's gone through circularization via Gibson Assembly, can be done on the machine. And this is just one additional step to the hands-free pipeline that scales up this very small number of molecules in the nanogram quantity that are circular and cloned, containing whatever number of synthetic DNA fragments you've decided to add, and scales this number up from nanogram quantities to about 10 micrograms of final DNA. So what that does effectively is remove the in vivo component of the scale up. So there's no overnight culture, there's no purification. This is DNA that's generated cell-free in microgram quantities, which can then be used directly in transfections or transcriptions as template. One thing that does need to be done, the DNA that comes off of the machine, the first step that the user would do is to digest it down into monomers, which is what this picture is showing. And from there, you're ready to go. So it's a significant time savings, and also another component that adds about 6 hours onto the runtime, but saves you the overnight scale up and the purification that needs to happen in vivo. So with that, I just want to pass it over to Krishna. I hope that now everyone has lots of great ideas on fast and effective ways to combat the virus. And Krishna will tell us all about how he generated the first fully synthetic SARS-CoV-2 genome.
Thanks, Katie. I'm going to start with a brief primer on the process of hierarchical assembly. This was developed by a team of scientists at JCVI, led by Dan Gibson. What I'm showing here are two prominent examples that demonstrate how hierarchical assembly has been successfully deployed to build synthetic genomes, completely de novo from oligonucleotides. The Mycoplasma genetolium genome, that's a little above 500,000 base pairs, and the Mycoplasma mycoides genome, which is about 1 million base pairs, were built using this process. Hierarchical assembly, in its literal sense, refers to the technology to enable building genomes or large complex DNA in stages of increasing complexity. The process of hierarchical assembly entails breaking the genome down to small overlapping pieces, sometimes up to 3 orders of magnitude compared to the size of the full-length genome. This essentially lends itself to the synthesis process from oligonucleotides. We refer to these as stage 1 molecules. These small pieces of genome could then be cloned into vectors and sequence verified before moving on to the next stage of assembly. The stage 1 molecules will then be stitched together to create stage 2 molecules, and stage 2 molecules will be assembled into larger stage 3 molecules, so on and so forth. This way, at each subsequent step of the assembly process, we increase the complexity of the DNA that's been constructed, specifically in terms of length. For example, the stage 2 molecules could be 2 to 10 times larger compared to the stage 1 molecules. This process is repeated until we assemble the entire genome as a single construct. Several technologies had to be developed in order to successfully deploy the hierarchical assembly process to build genomes. As one can imagine, we need a really good design software to be able to design the entire assembly process in stages to enable hierarchical assembly. The Gibson Assembly method to stitch DNA molecules with homologous ends is another wonderful outcome of the genome synthesis project. The flexibility to shuttle between bacterial and yeast host organisms and the development of transmission-assisted recombination cloning method in yeast were also important to enable the synthesis of large and complex DNA molecules, including genomes. Now, I'll provide an overview of how we used the hierarchical assembly process to synthesize the SARS-CoV-2 genome. We used our proprietary software to break the genome down to 24 overlapping 1.4 kb parts in silico. Again, these would constitute the stage 1 molecules. 3 consecutive stage 1 molecules were then combined using Gibson Assembly to generate stage 2 molecules, each approximately 5 kb in length. 6 of these stage 2 molecules were then combined again using Gibson Assembly to generate stage 3 molecule, which ended up being the final stage of the hierarchical assembly process, resulting in a roughly 30 kb genome of SARS-CoV-2. The stage 1 molecules were generated using the BioXP 3200 system. Like I mentioned earlier, stage 1 molecules consist of 24 overlapping DNA constructs that are about 1.4 kb in length. These molecules were designed and ordered through the BioXP portal, and once the reagents were available, we executed the run. This resulted in the generation of error-corrected and amplified 24 stage 1 molecules that span the entire genome of SARS-CoV-2 in a single overnight run. We then cloned the stage 1 molecules and obtained sequence-accurate clones, which were then used in subsequent stages of the hierarchical assembly. We combined 3 consecutive stage 1 molecules through Gibson Assembly method in order to generate 6 stage 2 molecules. These molecules were then combined again into the final stage of the hierarchical assembly, the stage 3 in this case, using Gibson Assembly method in order to generate the full-length genome. The genome was captured in a bacterial artificial chromosome at this final stage of assembly. We verified the presence of full-length genome in our clones using several methods. Since the genome that we cloned was fairly large, we used a technique called field inversion gel electrophoresis, or FIGE in short, to be able to resolve the cloned DNA at high resolution. We used restriction enzyme digestion of the DNA isolated from our clones to release the SARS-CoV-2 genome and analyzed this DNA using FIGE. This enabled us to easily visualize the cloned high molecular weight DNA. We also subjected the SARS-CoV-2 genome cloned in a bat to a supercoiled DNA analysis, which clearly indicated the size of the DNA that was cloned. This process involved resolving undigested full-length DNA alongside site standards to estimate the size of the DNA that was cloned. We then picked a few clones and identified a sequence-accurate clone using next-generation sequencing. Genome coverage across one such clone is shown on this slide. This concludes how we generated the full-length genome, and I will now move on to the applications of the full-length genome synthesis technology. Live attenuated vaccines are one of the most important applications of the full-length genome synthesis technology. There are several advantages of live attenuated vaccines, including robust and durable immune response. Boosters are rarely needed. It involves low cost and also generates really fast immune response. Attenuated vaccines have been extremely successful for all previously mentioned reasons, to provide immunity against viral diseases such as measles, mumps, rubella, smallpox, chickenpox, polio, yellow fever, and also against bacterial diseases such as typhoid and tuberculosis. The live attenuated vaccines technology already exists for influenza or seasonal flu. One can imagine that a similar technology would be developed for SARS-CoV-2 as well. There are several genetic changes that can be quickly generated within the SARS-CoV-2 genome. For example, inactivation of specific proteins could render the virus replication deficient or unable to correct the errors that occur during replication. These changes can then be successfully used to generate live attenuated versions of the SARS-CoV-2 virus, which can be used as a vaccine. The importance of the development of diagnostics for SARS-CoV-2 cannot be overstated at this point. With the full-length genome, we can assist the development of RNA or DNA-based diagnostics that span the entirety of the genome. These could be RT-PCR or next-generation sequencing-based assays. This is important because it enables researchers to pick any number of targets from any or all parts of the genome and successfully develop diagnostics. Not only that, our technology also allows for the rapid generation of diagnostics against specific genomic variants of SARS-CoV-2 as well. Some of the other applications of whole genome synthesis technology are highlighted on the slide. We discussed one aspect of vaccine development against COVID-19, but of course, there are multiple ways to train the immune system against this pathogen. All of these methods can be fully supported by the whole genome synthesis technology. Using full-length genomes, researchers are also able to choose from any number of antigens within the genome to target for the development of therapeutics as well, such as antibodies. Another important application would be using the modified versions of the full-length genome of SARS-CoV-2 to study the virus itself. For example, tagging different arcs with reported proteins can help us understand the life cycle of the virus during entry and infection. This concludes my section of the talk. I thank everybody for their attention. With this, I'm going to throw it back to Peter.
All right, thanks very much, Krishna. So while everyone is submitting their questions, I would like to highlight that in addition to the BioXP 3200 system, we actually have a number of products and services that we've recently developed, including the SARS-CoV-2 full-length synthetic genome that Krishna was just referencing. The full-length genome is actually available with or without a T7 promoter region, and additionally, we have RNA controls with overlapping fragments available for diagnostic assay development. Okay, and with that, I would like to go ahead and open up the floor for our Q&A session with Krishna.
Thank you, Dr. Gibson, Dr. Kannan, and Dr. Lyons for your informative presentation. We will now start the live Q&A portion of our webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. And now let's get started. Okay, our first question is, what role do you see Codex playing once the immediate threat of COVID-19 is past?
So, my name is Krishna Kannan. I'm happy to answer the questions. So, one thing that Codex has been focused on is developing rapid DNA writing technologies. And it's not just about synthesizing DNA molecules really quickly, but we also make really accurate DNA molecules as well. So one role that Codex can play once the COVID-19 pandemic is behind us, we can essentially enable the research community and scientists around the world to be able to be better prepared for pandemics like these. We are enabling distributed manufacturing in terms of DNA writing technologies, and we're going to come up with a whole suite of products as well, which is going to help the scientific community in a lot of ways to be better prepared for a future pandemic.
Thank you. And Dr. Kannan, what are some of your other COVID offerings?
So, Codex has several off-the-shelf products. You just heard how we synthesize the full-length genome. We have those genomes available off the shelf for researchers around the world to enable them to develop vaccines, diagnostics, and therapeutics. We also are very open to collaborating with people around the world to generate custom genomes, so not just the wild-type genome that's available off the shelf. We can make designer genomes for you if you're interested in trying out different designs to develop, for example, live attenuated vaccines. We have come out with RNA controls that are both overlapping and non-overlapping. So this way you get 100% coverage of the SARS-CoV-2 genome. And we also have library services that are available for use. And just come and talk to us. So visit us at codexdna.com. We have several products and services that are readily available for customers.
Now, Dr. Kannan, I'd like to hear your opinion on this. As a leader in the field, do you think the synthetic biology community has responded adequately to the ongoing pandemic?
I think we have done the best we can with the tools that are available. But what's really going to make a difference is new tools that would be developed, which will enable researchers to write DNA faster and get to their final designs much faster. And that's what Codex is trying to enable people around the world. It's distributed manufacturing of DNA writing that we want everybody to be able to use and generate their DNA. Without DNA, you're not able to do a lot of things, especially if you're in this field of trying to combat a pandemic like COVID-19. So hopefully once we have distributed manufacturing in biology, I mean, distributed manufacturing does exist for other fields, but not in biology, really. Codex is trying to make that happen. So that's what's going to make a difference when we encounter a future pandemic.
What do you think we could have done better to increase our general preparedness for future pandemics?
So this goes along the lines of the answer that I just gave. It's all about how distributed manufacturing can work in biology. So instead of having centralized manufacturing for your starting material, which is most likely the DNA that you use for research purposes, and if you want to develop vaccines or therapeutics or diagnostics, you're going to start with DNA. And just to the power of enabling this capability for researchers around the world, it's going to be extremely important and useful, and it's going to make us much better prepared for a future pandemic.
Thank you, Dr. Kannan. And we have some great questions coming in, and we have time for a few more, but I do want to remind our audience that any questions we're unable to answer today, and those additionally that are submitted during the on-demand period, they will be addressed by our speakers via the contact information you provided at the time of registration. Okay, let's continue. What is the turnaround time for submitting a sequence to final output?
So there are two ways of answering this. You're able to, we just brought our service option back, so you're able to order sequences off of our website, and we can synthesize these sequences for you, and the turnaround time generally depends on how complex and how big the sequence is. But if you're talking about the BioXP, if you have access to a BioXP or DNA printer, then once you input the sequence on our BioXP portal, within three to 4 days, you receive a reagent block from us, and after an overnight run, a single overnight run can actually produce 32 different 1.8 kb gene fragments for you. So within a week, you'll have access to this DNA.
Excellent, thank you. What other applications can this technology support beyond vaccine development?
There are several applications of DNA synthesis in general, and I'll speak to the whole genome synthesis technology. Just to be able to make modifications in the genome of a virus like this, which is very novel, and to be able to study the virus. And we've had several people approaches for making modifications in the genome, which include, okay, can we have reported genes in the genome, or can we tag different genes in the genome? And all this is going to help people is to study the virus itself, because it is a new virus and we are trying to understand how it works. That's going to be an important application as well, apart from the live attenuated vaccines, or in general other vaccines, the DNA, RNA vaccines, or viral vector vaccines, and other diagnostics or therapeutics that people can develop using the whole genome technology.
Dr. Kannan, can we order SARS-CoV-2 full-length synthetic genome itself from Codex, or do we need the BioXP system to synthesize our own synthetic genome?
You can do both. The SARS-CoV-2 full-length genome is available from Codex DNA. It's an off-the-shelf product that's available for our customers. We do a very careful screening of our customers, of course, to make sure that it doesn't end up in the wrong hands. Or you could get a BioXP from us, and you're able to synthesize parts of the genome and put it together as well.
Thank you, Dr. Kannan. And we have time for one more question. Let's go with this one. How many back clones do you need to sequence to get a SARS-CoV-2 sequence without any errors?
Like Katie described, we do have a proprietary error correction process, which enables the BioXP to generate really, really accurate DNA molecules. So we didn't have to sequence a lot of back clones to get to an accurate one. We did sequence 24, but we found a lot of positives in that. So it wasn't a lot.
Thank you, Dr. Kannan. And do you have any final comments for our audience?
Just one thing, visit us at codexdna.com. We have a lot of products that are off the shelf or that are custom solutions that might be available for you. Especially if you are in COVID-19 research, we just want to help people around the world to get through this pandemic, and we are putting our technology to full use to enable researchers around the world to develop tools to combat the pandemic. So visit us at codexdna.com.
Thank you, Dr. Kannan. And to our audience, you can also visit Codex DNA right now in our coronavirus event. I would like to once again thank Dr. Kannan, Dr. Gibson, and Dr. Lyons for their informative presentation, and Dr. Kannan for this excellent Q&A. I'd also like to thank our audience for joining us today and for their interesting questions. As a reminder, those questions we were unable to answer today, and those that are submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. One final thank you to our sponsor, Codex DNA, for sponsoring today's webinar. This webcast can be viewed on demand through the end of this year, 2020. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We hope to see you again soon. Have a great day.